The effect of plasmid copy number. One of the factors that can influence the expression of cloned gene is the availability of ribosomes in the cytoplasm. But this is not the problem because ribosomes, they are abundant in the cytoplasm, and they may be up to 15,000 ribosomes per cell, and they may represent up to quarter the dry mass of the cell. So another factor then that may be important is the availability of the messenger RNA transcripts. Because the number of ribosomes as compared to a particular messenger RNA transcript, it is comparatively high. So the availability of such messenger RNA transcripts, they can also determine the level of expression of the cloned gene. And the level of transcripts we can control further by increasing the dosage of the gene. That is, we can increase the copies of the gene within the cell so that we can get high level of messenger RNA. And in turn, it can be determined by cloning the target gene into plasmid that is having high copy number. That is, more copies of plasmids per cell. And in previous discussion or lectures, now we have the idea that there are different plasmids that can be used and have high copy number. And one of the example is artificially constructed PBR322 plasmid. PBR322 प्लाज्मिड को कंस्ट्रक्ट किया गया है, या दूसरे अल्फाज में हम ये कह सकते हैं कि ये डेरिवेटिव है एक और प्लाज्मिड का जिसको हम कोल E1 कहते हैं. और उम्मीद है कि आपको ये सारे प्लाज्मिड्स के नाम जो हैं, ये हमारे जो प्रीवियस डिस्कशन है, ये लेक्चर्स हैं, उनमें से ये आपको याद होंगे. तो ये जो कोल E1 plasmid है, it is also an example of high copy number plasmids. And this high copy number of plasmid, it is in turn is determined by the region of replication of the plasmid. Ori region, it is the region from where the DNA replication within the plasmid it started during the time of its duplication. So the region of replication, it can control the copy number of the plasmid. It will determine that particular plasmid, it will be low copy number or high copy number. And here two negative regulators, they can determine the copy number. And these two negatively regulated components are one of them, it is RNA molecule, which is synthesized by region of replication, and it is having the size of around 108 base pairs, and it is called as the RNA1. So it is one of the negatively regulated component. Another component, it is a repressor protein which is called as the ROP or ROP protein. So these two negative regulator components, they can determine the copy number of the plasmid by controlling the interaction of another RNA with the region of replication. And this example of another RNA, it is RNA2. This RNA2, it is also synthesized by a region of the DNA within the region of replications. Or in other words, we can say that the same region that synthesized RNA1, it is also synthesized RNA2. RNA2, it is having the size of around 555 base pairs, and it is complementary to the RNA1 because they are synthesized from the same DNA segment of the region of replication. So they are complementary with each other, but the strand for this synthesis, it is different. So by interacting with RNA2, RNA1, it can control the copy number of the plasmids. Now how it can control the copy number? For example, RNA2 if it is available and RNA1 if it is absent. So in the absence of RNA1, it can act as a primer with the region of replication, and it can start the duplication of the plasmid. But on the other hand, if RNA1 is available, which is complementary, and so that the conformation of RNA2, it is now changed and it is no longer able to act as a primer with the region of replication. तो इस तरह से प्लाज्मिड की सिंथेसिस नहीं होगी. और इसके साथ साथ अगर रिप्रेसर प्रोटीन भी है जिसको हम ROP प्रोटीन कहते हैं, तो ये जो RNA1 है उसके इनहिबिटरी एक्शन को ये ज़्यादा एक्टिवेट करती है. तो इस तरह से RNA1 और ROP प्रोटीन जो है आफ्टर इंटरेक्टिंग विद RNA2 molecule, it can control the copy number of the plasmids. If we have some plasmids that are deficient or mutant for RNA1, like in case of PAT153 plasmids, or even other plasmids that are mutant for ROP regulatory or repressor protein, even then the copy number of the plasmid, it can be increased. So by using the bacterial strain or the host strains that are deficient for RNA1 or regulatory protein like ROP protein, the copy number of the plasmid, it can be increased. लेकिन इसके साथ साथ हमें ये भी ध्यान में रखना चाहिए कि जो कॉपी नंबर है पर सेल, ये जो होस्ट की जेनेटिक्स है वो भी कंट्रोल करती है. Now different scientists, they have managed the copy number of the plasmids, like they have used two regions of replication that are working at different temperatures. For example, one of the plasmids, they have a region of replication that is activated at 30 degrees Celsius, and after interacting with the partition region, they can maintain the copy number at low level. When growing at low temperature, that is around 30 degrees Celsius. On the other hand, the other region of replication, it is activated at 36 degrees Celsius, so that the region of replication, now it is activated and it is having high copy number. So by managing the temperature, by using two regions of replication, some workers have managed the copy number of the plasmids. Under some circumstances, if mutations are there that can select the low copy number plasmids, then the high copy number plasmid, they may be phase out, and this problem can be tackled by subjecting the cells to antibiotics. On the other hand, if mutations are in such a way that low copy number plasmid, they are present one copy or two copy as compared to high copy number plasmids, like in case of PBR322, if low copy mutations are 1 in 50 plasmids, then it is not going to influence, because high copy number plasmid will maintain in the cell because of high rate of cell division.